Hi! Lab algorithm of gram-negative rods is a very high yield topic for both students and clinicians in practice. So I decided to make this video about the simplified algorithmic approach to the laboratory diagnosis of gram-negative rods. There are just possibly three major decision point questions to focus when we have gram-negative rods. One is to figure out if they are coma-shaped or not, and that includes the group of Campylobacter, Helicobacter, and Vibrio. If they are not coma-shaped, then we need to figure out what is their pattern of lactose fermentation. If they are fast, if they are slow, or if they are non-lactose fermenter. And the third decision point question is if they are non-lactose fermenter, we need to apply some advanced agar medium such as TSI agar to distinguish the major four pathogens, so-called Shigella, Yersinia, and proteus and Salmonella that belong to the non-lactose fermenters, while the fast and slow lactose fermenters, each one of them have a couple of their well-known differential diagnoses that we will review. So let's dig deeper into this and simplify this high-yield algorithm for you. If you have a gram-negative rod, the first thing to do is to figure out if it is coma-shaped or not. The three coma-shaped enteric gram-negative rods, or just so-called coma-shaped gram-negative rods, could be distinguished based on two factors: their growth pattern and requirements, as well as their motility. If it is Campylobacter jejuni, their growth requirement is defined to be microaerophilic, which is 5% oxygen, 10% CO2, and temperature as high as 42 degrees centigrade, because they cannot grow at temperatures lower than 25 degrees. If it is Vibrio, they require alkaline environment for their growth. And finally, if it is neither one of these two categories but is said to be a urease positive coma-shaped enteric gram-negative rod, that's H. pylori. On the other approach, we can think of their motility pattern. The motility of Campylobacter jejuni is defined as darting motility, while that of Vibrio is defined as shooting star motility. And of course, like in everywhere else, we can have their clinical syndromes. You know, Campylobacter is associated with acute gastroenteritis, usually bloody diarrhea, especially among children and young adults. Vibrio is associated with non-inflammatory, non-bloody, but watery diarrhea, which could be very severe, and you hear the word rice water. And H. pylori is associated with four major categories, especially gastritis, peptic ulcer disease, erosive gastritis of acute type, as well as a couple types of gastric cancer, such as adenocarcinoma and maltoma. Among them, always remember, only H. pylori is urease positive. It's logic, isn't it? It helps it to survive the stomach acidic environment by producing ammonia, alkaline environment around itself. So let's move on to the real juice and meat of this discussion, which is the categorization of these enteric gram-negative rods based on their lactose fermentation. Remember these differential diagnoses. If an enteric gram-negative rod is said to be fast lactose fermenter, it's either Klebsiella or E. coli or Enterobacter cloaca. If it's said to be slow lactose fermenter, it's either Serratia or Citrobacter or one of our coma shapes, which is Vibrio. On the other side, if it is non-lactose fermenter, it's either Shigella, Yersinia, or it is proteus, Salmonella. Exactly as I am mentioning it. Shigella, Yersinia, or proteus, Salmonella. You will see why. Okay, so if they are fast lactose fermenters, how are we going to distinguish them? Well, if they are motile, it's either E. coli or Enterobacter cloaca. If it's non-motile, it's going to be Klebsiella. Now, of course, you know that Klebsiella produces this thick capsule. However, remember that capsule per se is not enough to distinguish them, because E. coli also has capsule. It's motility. Most importantly, it's motility. And remember, E. coli is indole positive. So if it is not indole positive, if it doesn't have thick capsule, and specifically if it is associated with nosocomial urinary tract infection, it's neither E. coli nor Klebsiella, because E. coli has motility and capsule and is indole positive. Klebsiella is urease positive and has thick capsule and is non-motile. So therefore, if none of these requirements are met, it's going to be Enterobacter cloaca. Final group before the main discussion of non-lactose fermenter is slow fermenters. What does it mean if an enteric gram-negative or gram-negative rod in general is referred to be as a slow fermenter? It first of all means slow fermenter of lactose, and secondly it means that it doesn't ferment lactose as fast as the fast fermenters in the routine environments. However, if we give it enough time or if we use different environments, they may be able to ferment lactose. So how we distinguish them? Well, if it is common shape, it's Vibrio. If it produces red colonies, it's Serratia marcescens. If it's neither one of those, it's going to be this bug called Citrobacter. Finally, the important group, the non-lactose fermenters. These are either Pseudomonas, which is the only one that's oxidase positive, or they are going to be the ones that we use the acronym SHIPS for them. Shigella Yersinia, Proteus, Salmonella. You ready for that? Remember, if a non-lactose fermenter enteric gram-negative rod is oxidase negative, it is SHIPS, Shigella Yersinia and Proteus, Salmonella. And for us to distinguish them, we need to use TSI agar. What is this TSI agar? TSI agar, or triple sugar sulfate iron agar, is an agar medium with a phenol red pH indicator that will turn yellow if there is enough fermentation to cause acidic environment formation. Also, it has this sulfate that could react with iron and produce the thing that is called black dot or eyefish. Again, black dot or eyefish is formed if the bug is capable of producing H2S and therefore combining it, the sulfate group, with the iron in the environment, producing ferric sulfate, and that produces this black eyes or black dots. And so based on that, we are going to categorize these 4 pathogens. First, 2 letters, SH and Y. I call it 2 letters for Shigella and Yersinia. They are the ones that don't produce H2S and therefore they lack production of the black dot or fish eye in TSI agar. How are we going to distinguish these 2 from one another? That's based on their clinical presentation. You know that Shigella is associated with dysentery, especially among certain demographics such as male homosexuals or residents of nursing homes or children. On the other side, Yersinia enterocolitica is associated with mesenteric adenitis, a syndrome that's sometimes mistaken with appendicitis and therefore is referred to as pseudoappendicitis, and also it can cause enterocolitis. Don't confuse Yersinia enterocolitica with Yersinia pestis, by the way. Also, remember, both of these 2 non-lactose fermenters that do not produce H2S are also non-motile. So there are triple non. They are non-motile, they do not produce H2S, and they are non-lactose fermenters. However, the other group, not SH and Y, not Shigella Yersinia, but PNS, the so-called Proteus and Salmonella, they are motile and they produce H2S or gas in the TSI agar. So always use this SHIPS acronym to memorize this most high yield part of this entire lecture. SH and Y, PNS. And also you can think of it this way. A ship is usually don't move initially and then it starts moving. In this case, not only they move the last 2, which is PNS, Proteus and Salmonella, but they also produce some gas. So they are the motile one that produce gas. Now, if you still have something left in your brain to handle this, you need to want to know how do we distinguish Proteus from Salmonella, and even further or deeper, how do we distinguish different Protei from one another and different Salmonella species from one another, based on just this lab algorithm, not talking too much clinically. Remember, among all Salmonella, only Salmonella typhi is H2S negative, which is probably the most counterintuitive or paradoxical fact to mention in this entire lecture. It confuses the distinction with the other group, the SH and Y, which are also H2S negative, while the non-typhi Salmonella are H2S producers. That said, how do we distinguish the different Proteus pathogens from one another? Remember, among the Proteus pathogens, Proteus mirabilis is non-indole positive, while all the other Proteus bugs which are more pathogenic are indole positive. Now, how do we distinguish proteus from salmonella? Is very easy. Proteus is urease producer, salmonella is urease negative. Don't memorize that, just remember that Stackhorn calculi, the renal stones that are formed in the alkaline environment because of proteus, that is similar to that of H. pylori, that produces alkaline environment around itself to protect itself from the acidic environment. Proteus produces alkaline environment, predisposes to formation of Stackhorn stones. Both of them are urease positive. So once more, remember the whole algorithm. If you have gram-negative rods, figure out if they are coma-shaped. If they are coma-shaped, it's H. pylori, Campylobacter, Helicobacter. If they are not coma-shaped, figure out if they are fast lactose fermenter, slow lactose fermenter, or non-lactose fermenter. If they are non-lactose fermenter, apply TSI, agar, and oxidase test to figure out if they are pseudomonas, if oxidase positive, or if they belong to the ships. If they move and they are motile and produce gas, they are proteus and salmonella. If they don't, they are Shigella and Yersinia. And that is the gram-negative rods. Thank you.